Good morning, everybody. In the theme of the three of us here, you can tell us easily apart. I'm the one with the longest hair, and certainly the youngest outlook. So, moving on. It's what have these two gentlemen left me to take on in the great tradition of success of DNDi. And it's incredible what's been done in 10 years. And for the next 10 years, here I am to try and put us on the right road and continue this great success. So what I'd like to do is describe what's come out of the short, medium, and longer term strategies. And here's the first summary of that particular effort. This is our portfolio, and what you can see on the top from left to right is the research, translation, development, and implementation stages, and on the y-axis there are our diseases. And you can see we have a few diseases, but what's significant in this portfolio is that, first of all, we started to bring through new chemical entities. Those are the boxes with stars. We're still trying to bring improved therapies to meet immediate patient needs in later development stages, such as these combination therapies you can see in the disease areas. And we have 6 new treatments. All of these different phases raise different challenges for DNDi. And we aren't done when we've actually just registered the drug. There's still things to be done. Now, what I'd like to describe is some of the challenges we have, some of the opportunities in that final mile, making sure that the pill comes to the patient. And one of the things that we face with all of our drugs is to make sure that we have a sustainable supply. And in the case of pediatric benznidazole, we're still trying to ensure that we have a second manufacturer. And we found that's very important because of political instability. Maybe there's a manufacturing glitch, maybe something else happens, the ship sinks, or in the case of one of our suppliers, the ship is set on fire and we lose the machine. So we need to think of these possibilities, and it's kind of taking me out of where I used to be in pure R&D terms and putting me into a world that's quite challenging to think about in terms of all the things that you need to ensure to be successful. And we also need to make sure that as we are more successful, and in the case of benznidazole for adult use in chronic Chagas, where we've had a very, very good trial performed by Isabella and her crew, showing that benznidazole is a very effective, safe therapy over a prolonged period of time for Chagas patients, that we actually communicate this well and we advocate well, and that we inform health systems and governments that they need to think carefully about using this drug in a different way from what they've done in the past. And we need to keep putting those messages across so that we consolidate and sustain the use of our drugs that are much better than the old arsenicals, much better tolerated, and in most cases showing much greater efficacy. Similarly, in the case of leishmaniasis, we've made great strides there, and the big challenge for us now is to make sure that the available combinations that are much better than they were 10 years ago are taken up and provided through the health systems for the patients. And for this, we really need to keep working hand in hand, not only with endemic countries, but making sure that our disease platforms that we've built for most of the major diseases, that these are used effectively to ensure that goal. So moving on from there, the next question is what are once we've made sure that our therapies we have today are available and can get to the patient, what can we do to improve existing therapies to meet immediate needs? And here the challenges are somewhat different. The challenges are different because we have to bear in mind that the diseases we work on are caused by infectious organisms. And they've had millions of years to co-develop with us, to co-evolve with us. And we think we know them well. I think they know us better than we know them. And so there are still challenges, and we're still learning lots of things. And a good example of that is benznidazole for Chagas disease. I've just told you that we've had a great result from a clinical trial. This is good because it's clear data coming from a well-designed trial, so we can rely on it. By analyzing that data carefully, it gives us ways to go ahead with benznidazole, which is a tricky treatment. It's effective, but it comes on longer-term dosage with some side effects, and some of those side effects are not what we would like to have in a perfect compound. But analyzing that, I think we have a way forward to make this a better drug, meaning better tolerated. And the data is telling us that we have a way to think about short-course treatment of benznidazole. Similarly, for leishmaniasis, although we've been incredibly successful on the Indian subcontinent, we have not been as successful, I would say, with disappointing results in Africa. The combination approach we've taken there has not worked to the same levels of efficacy that you would like to have. compared to India. And that's a challenge. We need to look at our data carefully and think carefully about drugs we may need to use in the future based on the biology and the clinical trials, what it informs us to do next. And we also have to cope with new manifestations of the disease, such as HIV, VL. Here we have HIV, which suppresses the immune system, VL, visceral leishmaniasis, that suppresses the immune system, and together they're making a syndrome of the disease that's incredibly difficult to treat, and we have to think through new ways of how we would develop medications to do that. Similarly, in areas where there's unmet medical needs, such as pediatric HIV, we have some challenges. Simon already alluded to the fact that making a fixed dose combination isn't as straightforward as you would like, and I think pediatric HIV is one that shows you maybe the challenges are not where you expect. So in pediatric HIV, the first challenge we had is how do we define the dosage for babies through to 6-year-olds? It's not as straightforward as taking the weight of adults and dividing it by the weight of a child and coming up with a factor. So that's one particular approach. And as we know, we need to make combinations. These are very successful at extending the lives of adult patients with HIV. We need to have an effective combination, and the delivery route in children, the easiest delivery route is to give to food. And so our main challenge here is to make a taste-masked 4-in-1 combination. Easy to say, difficult to do. And if anyone's ever tasted these drugs, then I would encourage you, if you ever come to DNDi, to please take part in an open tasting. These taste really horrible. One little bit and you have it for 24 hours in your mouth. So these poor children today who were fed these drugs, if you see the videos of them, they're on the website, they clearly don't like it, and I can understand why. We can do a lot better than that. It's a technical challenge here as well as a biological challenge to bring forward that new therapy. And we need to keep pushing on our other work in sleeping sickness with drugs that you've already heard about. Beyond that, I think the ultimate, 10 years from now, I'd really like to see a change in the practice of medicine. And what I mean by that is that we can actually go into a village, a community, we can test people in that community for our diseases, and we can give them a pill, very much like an aspirin, it would be the ideal, that would remove their pain or cure their illness, no side effects, and we can walk away, job done, we've done a great, we've met the need here. I think in sleeping sickness, we're merely making rapid advances, and I'm hoping that by 2016, that fexnidazole may register as a drug for sleeping sickness, our first orally available brain-penetrating drug that will cure stage 2 and hopefully stage 1. And behind that, just in case something does go wrong with Phexi, and we're hoping it doesn't, I'm gripping onto wood over here as a good luck charm. We have 7158 in case we need other options. And as Simon said, options are always really good, and I'll come back to that theme in a minute. In Chagas and leishmaniasis, we really have to work hard over the next few years to find new compounds. New compounds that are orally active and work in different modes from our old compounds, from our old therapies. And one of the examples of that new generation is VL2098. Not only is it orally active, not only is VL2098 orally active, but it's in animal models, when we treat animals with that have been infected with leishmaniasis, has a very interesting additive feature, and that is, compared to other drugs we've tested, it causes sterile cure, meaning that we can look at the animal tissues following treatment and not be able to find any evidence of the parasite being there. That's a very interesting feature, because up till now, most experts will tell you if you treat and you can reduce the parasite to a certain level, the patient will cure themselves because their immune system is able to kick in and then carry on the curing of the disease. And that's a kind of a unique feature. If we could engineer that into our compound, we would give the person's immune system even more help to rid themselves of infection. And we are still carrying out near-term patient need approaches in the area of filarial diseases, helminth infections, where we're looking to repurpose drugs from pharmaceutical and animal health libraries as a quick way to get to meeting patient needs. Now, what's the future? I really think we need to access and improve on diversity. Having options because of high attrition rates in drug development is extremely important. And up until now, DNDi has only been able to access a small selection of chemical diversity, which is what makes you new compounds, brings you different drug development options. The universe is much, much bigger. And we all, those of us who were here at the scientific symposium yesterday, heard about some of the ways we can do that. We have to work really hard on accessing that chemical space. And we also need to innovate in how we translate into the clinic. I think we need to think differently about our diseases, less about the classical phase 1, phase 2, proof of concept, and then phase 3 registration trials, and try and think of adaptive designs and moving rapidly out of phase 1 when we know that our drug is well tolerated and is exposed in the person's body, to finding out if they're going to work, and then moving that as quickly as possible to a result that will hopefully convince regulatory authorities to license the drug for use. Those are two immediate things that I think over the next couple of years we need to do a lot of thinking about and innovation on at DNDi. We also need to tap into the vast resource in the endemic countries. We haven't so far done that to a great extent, and I think it's something that over my last 18 months since I've been here, is a vast untapped source, and I'm hoping over the next 2 years we'll start to see outcomes from those investments. And then last but not least, we need to think of different modes of doing our drug discovery, of fueling the engine. And up till now, what you've heard about from Xing and Simon is the approach we've used, which has essentially been bilateral, where we've looked around for partners who can help us, or partners have approached us to help them to take drugs into the clinic and show that ideas work. And you can see here that we've actually got we have a lot of bilateral partnerships. The drawback with that is that the partners and we don't tap into cross-seeding that would come from where we would have multilateral relationships. And this would really help us because it's the knowledge that everybody has, as well as the resources they have, that really would give you a big leg up into getting to new chemical entities that would be effective as therapies. And we're hoping, and I hope maybe in the next year or so, we can come back and tell you we've managed to achieve this. This is not a particularly new model. I hope it'll be a new flavor, especially in our space, in neglected tropical diseases. We're building on efforts that have been done for malaria and TB, in order to come up with a similar accelerator, booster of drug discovery for DNDi. So, where do I see, and this is a bit of a crystal ball exercise, to some extent, and to some extent I'm pretty much sure we'll bring some of these drugs. Are we on track to deliver for our business plan by 2018? Are we going to satisfy the expectation of our donors? Are we going to help the patients? They're the ones, they don't really care what the drug is, they just want a cure for their illness. But is it realistic we can bring new treatments for these diseases? And I think we can. We're going to make significant progress in sleeping sickness. I think we'll bring up at least a couple of new therapies for leishmaniasis, and certainly for pediatric HIV, I'm expecting to see big progress. For Chagas, there may be opportunities on the horizon, and for filarials, we're in an early phase. It's difficult to predict right now, but as soon as we have good compounds, we'll push very hard on moving forward. So looking from that, my penultimate slide is this one, preparing for the future. We're now entering a new business plan cycle. We will continue with DNDi for the next 10 years, and it will be successful, or over my dead body, I guess, or maybe not. These two guys still look alive and well, so I think I've got a good future. We have to revise our business plan, make sure that we're doing the right things and aligned with all of the various stakeholders. And we need to make sure that patients are at the core of our mission. It's the patients we're here for, and not everything else we tend to sometimes get focused on. And with that, I'd like to thank all of our partners and donors. There are lots and lots of people out there. There are some really significant partners. We've mentioned a few of them, and I'd like to thank all of you. Without you, none of this is possible. I wouldn't be able to stand here today and tell you about what we'd like to do over the next 10 years without that input. So thank you very much.